Okay, so that's kind of the deal with the DNA. Now, here's where things get fun. DNA has to replicate. It has to copy itself. So for simplicity's sake, we've drawn that sugar phosphate backbone in a straight line, and then these little things sticking out are the A's and the T's and the C's and the G's. Okay? And we're going to look at and add some molecules of DNA replication. So DNA replication occurs in S phase of the cell cycle. So anytime a cell needs to divide and copy itself, it's going to replicate its DNA first, and then something else and then it's going to divide. Okay, so it has to replicate its DNA first, and then it gets to divide. So there's a couple of lots of molecules that are involved here. The first one is we're going to draw it over like this. Okay, and this is step 1 here, and I'm going to kind of color code as I go. This is helicase, and helicase unwinds the DNA. Okay, so helicase unwinds the DNA. And then in step 2 is DNA wants to stick together. These things want to hydrogen bond to each other. The A's and the T's want to stick together. They have an affinity for each other. So there's actually some proteins that will stick along the back here, along the backbone. And that's going to be step 2. And those proteins actually have the easiest name, I think, to remember, because they are single-stranded binding proteins. They help DNA stay as a single strand instead of double strand. Single-strand binding proteins. They stabilize the unwound DNA. Okay. And then step 3 is we're going to have a molecule that you actually we're not going to draw it in, but we're going to have it because we're going to draw in something else here in a second. Step 3 is primase. Another enzyme, is going to add RNA nucleotides, and we'll talk about why in a second. Why RNA, not DNA? Because we're trying to make DNA, right? To the exposed bases. And so it base pairs. So that what that means is if it reads an A, it's going to put down a T. It's actually going to put down a U. But if it reads a G, it's going to put down a C. Okay? So we're going to have a little bit, and I'm going to do like a 3. Okay, that is my RNA primer. Okay, so that's my RNA primer that my DNA put down. Okay, then in step 4, a molecule called DNA polymerase 3. Don't ask. Okay, so this is step 4. DNA polymerase 3. We're going to meet 1 in a second, and you'll never ever meet 2. Don't ask why. Well, I can explain it to you, but you don't really care. It adds DNA nucleotides to the RNA primer. The reason why you had to add this RNA primer is that DNA polymerase 3 can't just start DNA replication. It actually takes a little bit for it to grab on and bind to that single-stranded DNA. So it actually has to have something to bind to it. So it actually has to have this primer to bind to. It's just not that cool that it can actually bind by itself. So it has to have that RNA primer in order for it to bind and then start. And all it's going to do, it's actually a really cool molecule. It's going to basically read an A, put down a T, read a G, put down a C, read a C, put down a G. Okay, just on and on and on. The only other thing is that DNA polymerase 3 has a little issue with is it only adds in the 5' to 3' direction. Okay, so this is actually going to be this strand's up here. It's going to be its 3' end, and this one will be its 5' end here. So it's going to go and synthesize in this direction. Okay, so and then the next thing that's going to happen is I'm actually going to write it in pink because it's kind of very similar to what happened in step 3. We don't want that RNA to stay there. We want to get rid of that RNA primer. So DNA polymerase 1, and I'm not going to draw it, is going to come in, and it's going to basically bind here to where the DNA and the RNA are bound together, remove the RNA, and put down real DNA. Okay? Is going to replace RNA primer with DNA. Okay, now, what we've done so far now, if we come down to this one, we're going to kind of just get rid of all that stuff. So I had, this was my 3' end, and this was my new, this is my new 5' end, and I've made this entire piece. So helicase is unwinding, I have my binding proteins binding, and my DNA polymerase is just cruising along. And as helicase unwinds right here, it's going to cruise along and read an A, put down a T, read a G, put down a C, read a C, put down a G. You're catching on, okay? And it's just going to go in that direction, in the 5 to 3 direction. The only issue is, and okay, let me just pause myself here. That is what we call our leading strand, okay, is what I just described. So we're going to kind of make that step 6, even though basically just summarize steps 1 through 5, is my leading strand. It's going to go down, it's going to proceed continuously, or it's produced continuously in the 5 to 3' direction. Five prime to 3' direction. Now, here's my issue. Down here on this strand, this one is my 5' one, right? So that means that the new strand is going to be 3 up here, but DNA polymerase 3 can only go from 5 to 3. So it's going to go in this direction. So we can actually have it, do it in, so it's going to lay down in this direction. That's the way that DNA's polymerase 3 is going. So it's going to actually make little fragments of DNA. Okay, little fragments. And so as helicase is winding here, polymerase 3 will bind, actually primase will bind, lay down an RNA primer. Polymerase 3 will bind, put down some nucleotides until it reaches this fragment, and then it'll fall off. And then meanwhile, there's now more exposed as helicase unwinds. So more primer is going to be laid down than DNA's polymerase 3. So we're going to end up with these little fragments. So let's just write what I just said. Okay. So that's actually the last step, step 7. Okay. So those are produced, and this is my lagging strand. It lags behind, it's produced in pieces. And those pieces get a really fun name. They're called Okazaki fragments, and they're obviously named after the scientist that discovered them. Okay. And many RNA primers are required for this event to occur. Okay, because I need a primer to start this piece, and I need a primer to start this piece, and I need a primer to start that piece. And then the primers have to be removed and replaced by DNA polymerase 1, just like before, but now I have lots of these primers that have to be removed and replaced. Okay, now the only thing is I have these little fragments, and the fragments, that's kind of the last step, is there's a new molecule that has to come in and fuse, and this is ligase, okay? And ligase is going to fuse those fragments together to make one continuous piece of DNA. He's going to fuse that sugar phosphate backbone together. Okay, so that's kind of the final step, is ligase will fuse the fragments together. So you think of it kind of like glue. One last thing about this, every time you add a nucleotide, it actually comes in not as just wanting with one phosphate attached. It actually comes in with 3 phosphate attached, so it actually comes in as GTP. And then those phosphates, 2 of them get hydrolyzed off. And so that's the energy that's being used to power this entire process. So it's a GTP or an ATP or a CTP that comes in and does it. So it takes a lot of energy to copy your DNA.